Hello, we shall be studying on the topic translation or protein synthesis. Translation can also be called as the protein synthesis. Now, what do you mean by translation? Translation refers to the process of changing the language of the genetic information that is being stored in the mRNA after transcribing the DNA into the amino acid. So the genetic information that is being stored in this mRNA is being changed into the particular amino acid. So this changing of the language of the genetic informations to a particular amino acid is referred to as translation. We shall be discussing on the mechanism of this translation. The mechanism of this translation involves various steps. The first step is activation of the amino acid. Amino acid, I've indicated amino acid with AA. Now, the amino acid in the cytoplasms occur in the inactive form, so they have to be activated. So this amino acid, when it binds with ATP, that is adenosine triphosphate, it gets converted into amino acyl adenosine monophosphate and the enzyme. So these conversions of the amino acids into its activated form is being carried out by an enzyme called as the amino acyl synthetase. So once the amino acid gets attached to the adenosine monophosphate and the enzyme complex, this form the activated amino acid. So this amino acyl adenosine monophosphate with enzyme complex can also be called as amino acyl adenylate enzyme complex. And since there is a triphosphate, so the remaining two phosphate are removed as a pyrophosphate. Now once the amino acid is activated, second step is that this activated amino acid have to be attached to the tRNA. Here tRNA refers to the transfer RNA. So the activated amino acid, that is amino acyl adenylate enzyme complex, is attached to the tRNA molecule to form the amino acyl tRNA, and the enzymes and the adenosine monophosphate are removed. So the amino acyl tRNA is also called as charged tRNA. Now this charged tRNA is used as an adapter molecules for decoding the genetic information that is being stored in the mRNA. Hence this charged tRNA is also called as the adapter RNA. So the amino acid will attach to the 3' of the tRNA. So this is the tRNA, this is the anticodon site, and this is the 3' So the amino acid will be attached to the 3' Hence, if the amino acid is attached to the tRNA, then the tRNA is called as the charged tRNA. So if the tRNA doesn't have this amino acid on its 3' then those tRNA are called as the uncharged tRNA. And if the amino acid is attached to this 3' RNA, but if the amino acid attached is of incorrect amino acid, then the tRNA are called as the mischarged tRNA. That means the amino acid is attached to this 3' RNA, but the incorrect amino acid is attached to this 3' of the tRNA. So such type of tRNA are called as mischarged tRNA. Now next we shall be discussing on the third step, that is the formation of the polypeptide chain. Polypeptide, poly means many, peptide here we are referring to the amino acid, so many amino acid. In prokaryotes 70S type of ribosomes are found. These 70S ribosomes are further divided into two subunits, that is the smaller subunit is called as the 30S subunit and the larger subunit is called as the 50S subunit. During the initiations of polypeptide chain, three initiation factors, that is IF1, IF2 and IF3, comes and bind to the smaller subunits of the ribosome, that is the 30S subunits of the ribosomes, with GDP attached to the IF2, that is IF2. So the GDP is found attached to this initiation factor 2, and these together they attach to the 30S ribosomes, that is the smaller unit of the ribosomes. Now the initiator tRNA along with its amino acid and the mRNA binds to the smaller ribosomal units, that is a tRNA. In bacteria such as E. coli, this initiator tRNA are also called as the formilatable tRNA, as represented by tRNA F-MET. And this formilatable tRNA in the E. coli carries N-formal methionine. Therefore, the initiator tRNA in E. coli are represented by F-MET. F stands for formyl and MET stands for methionine, and F-MET tRNA F-MET. So this is the initiator tRNA in case of E. coli. Now, the initiation factors, that is the IF, together with the GDP, the mRNA and the 30S subunits of the ribosomes, they together form the 30S pre-initiation complex. So this is the smaller subunit of the ribosome, 30S. So this is the mRNA and this is the tRNA together with its F-MET and the initiation protein, initiation factor with the GDP. So this tRNA will be joining to the P sites of the 30S ribosomes. These ribosomes have two sites, P site and A site. Now the P site is also called as peptidal site, and this P site will be carrying out the functions for attachment of the tRNA. Now these 30S ribosomes together with the mRNA and the tRNA with F-MET, they together form the 30S pre-initiation complex. Now this 30S pre-initiation complex that is formed joins with the larger subunits of the ribosome, that is 50S, and they generate the complete 70S complex ribosomes. And this is the 50S, this is the 30S. So joining of this 50S large subunit with a smaller subunit 30S to form a complex 70S ribosome is being the energy for joining this larger and smaller subunits is being provided by GTP. That is being attached to the initiation factor 2. So this is the smaller subunit, this is the larger subunit, this is the A site, this is the P site, and this is the mRNA, and this is the tRNA with its form methionine. Now after the formation of the complex, the next step involves the elongation of the polypeptide chain. After the formation of these 70S ribosomes, mRNA, F-MET tRNA complex, the elongations of the polypeptide chain begins by addition of the amino acids. Now the larger subunits, that is the 70S ribosomes, the larger subunit of the 70S ribosome, that is the 50S, has two slots, that is the P site and the A site. The P site is also called as the peptidal site or the donor site, and the A site is also called as the amino acyl or the acceptor site. The incoming amino acyl tRNA, so tRNA with the amino acid is called as the amino acyl tRNA. So the incoming amino acyl tRNA complex gets attached to the acceptor site, that is the A site of the ribosomes, and they base pair with the mRNA. So if the mRNA is carrying the nitrogenous base AUG, then tRNA will carry the complementary base pair of this. That means it will carry the base that is complementary to this AUG. So A always pair with T, but in case of RNA it will be U. So A with U, U with A, G, C. So this tRNA will be carrying the complementary base sequence of this mRNA. So this is how the base pairing of the tRNA with the mRNA is carried out. Now this is the first amino acid. Now the incoming tRNA with the second amino acid will also be attaching to the A site of the 70S ribosomes, and this, if this is carrying, this mRNA is carrying CGG, then this side will carry G, since C always pair with G, G always pair with C, G with C. So this will also be carrying the same amino acid as this. So this is how the base pairing is done. And now the two amino acids are joined together by the formation of a peptide bonds. Now how the two amino acid, this 1 and 2, once these two amino acids transfer, once this tRNA come and join here, the two amino acids will be joined together by peptide bond. So since this is the first amino acid, the carboxyl group of this amino acid will be joined by the amine group of the second amino acid. So this will be joined with this, the first amino acid will be joined with the second amino acid from the carboxyl group of the first amino acid, and the second amino acid will be joined to the first amino acid through the amine group. So this is how the attachment of the amino acid is carried out. So this is the complete ribosome. So these are the tRNA which is coming and pairing with the mRNA, and this mRNA, if it is carried, these are the nitrogenous base sequence of the mRNA that is carrying the genetic information of the DNA that is being transcribed from the DNA. So these are the nitrogenous base sequence. So this tRNA will be carrying the same complementary base sequence of this. So these are the amino acids that are formed by the ribosomes, by the movement of the ribosome along the entire length of the mRNA. So this is the first this is amino acid joined with the subsequent amino acid, and this is the tRNA with the fifth amino acid. So this also will be joined with this two peptide chain, and the incoming amino acid coming tRNA will also be base pairing with this, and the amino acid will be joined with this. So this is how the long chain of the amino acid is formed. Now after the formation of amino acid, the long chain of amino acid, the next step involves the termination of the polypeptide chain. So the elongation of the polypeptide chain is terminated when the A sites of the ribosome reach the termination codons. There are three termination codons, that is UAG, UGA, and UAA. So the tRNA cannot recognize this termination codon. Therefore, three releasing factors are released: RF1, RF2, RF3. So these three releasing factors are released in the prokaryotes, and they will recognize the termination codon. RF1 recognize UAA termination codon and UAG. RF2 recognize UGA, and RF3 will stimulate the function of these two. So this is how the amino acids, the long chain of amino acid that is formed, are terminated by this releasing factor, and this long chain of amino acid are called as the polypeptide chain.